Welcome back to Clinical Hematology Reviews. Today we'll be discussing blood composition, understanding how peripheral blood is structured at both the macroscopic fluid level and the cellular level is the absolute foundation for everything we evaluate in the clinical hematology laboratory.
Absolutely. Whole blood is a remarkably complex heterogeneous fluid consisting of a liquid matrix and suspended cellular elements. When we centrifuge an unclotted, anticoagulated whole blood tube, density forces the sample into three distinct measurable layers. The top liquid layer is plasma, which constitutes approximately 55% of the total blood volume. It is a clear to translucent yellow fluid containing water, crucial proteins like albumin and coagulation factors, as well as electrolytes, nutrients, and metabolic waste products. At the very bottom sits the red blood cell layer, packed with erythrocytes, making up roughly 45% of the total volume.
Sandwiched right between those two major layers is a very thin, whitish interface called the buffy coat, which makes up less than 1% of total blood volume. That narrow buffy coat holds virtually all of our circulating leukocytes, or white blood cells, and thrombocytes, which we know as platelets. It is also critical that laboratory professionals clearly distinguish between plasma and serum specimens.
That distinction comes down to coagulation proteins. Plasma is obtained by centrifuging anticoagulated blood, meaning it retains all active coagulation factors, including soluble fibrinogen. That makes plasma the required fluid specimen for hematology and hemostasis testing. Serum, on the other hand, is obtained after allowing whole blood to clot completely in a specimen tube without anticoagulants. During clot formation, fibrinogen and other labile clotting proteins are entirely consumed within the fibrin matrix. Consequently, serum lacks functional coagulation proteins and is primarily utilized for clinical chemistry and serology testing rather than coagulation studies.
Moving from the fluid matrix to the cellular components, let's explore the mature leukocytes circulating in peripheral blood, starting with the segmented neutrophil. The segmented neutrophil is the predominant white blood cell in healthy adult peripheral blood, accounting for a relative concentration of 50 to 70% of total leukocytes. Morphologically, mature neutrophils feature a distinct nucleus segmented into 3 to 5 distinct lobes, connected by fine, thread-like nuclear filaments. Their cytoplasm is abundant and filled with pale pink secondary or specific granules, and they measure approximately 3 to 4 times the size of a normal mature erythrocyte.
Clinically, the segmented neutrophil acts as our primary cellular defense against acute bacterial infections through rapid chemotaxis and active phagocytosis. When evaluating patient results, we routinely calculate the absolute neutrophil count, or ANC, by multiplying the total white blood cell count by the relative neutrophil percentage. For instance, if a patient presents with a total white blood cell count of 10 times 10 to the third per microliter and an 80% relative neutrophil count, we multiply 10 times 10 to the third by 0.80 to arrive at an absolute neutrophil count of 8.0 times 10 to the third per microliter. Standard adult reference ranges for total white blood cell counts typically span 3.6 to 10.6 times 10 to the third per microliter.
Transitioning from innate cellular defense against bacteria to adaptive immunity, let's discuss the lymphocyte. Lymphocytes are smaller leukocytes characterized by a round or slightly indented nucleus containing dense, heavily clumped chromatin. They exhibit a high nucleus to cytoplasm ratio, meaning the nucleus takes up most of the cell, surrounded by a thin rim of agranular, pale blue cytoplasm. Because a resting lymphocyte nucleus is naturally equal in size to a normal mature erythrocyte, evaluators frequently use normal lymphocytes on blood smears as an internal visual ruler to assess erythrocyte size and detect microcytosis, smaller than normal red blood cells, or macrocytosis, larger than normal red blood cells.
Functionally, lymphocytes mediate adaptive immune responses and serve as the primary responders against viral infections. In healthy adult blood, lymphocytes represent the second most abundant white blood cell type, with a relative reference range of 20 to 40%. However, a vital diagnostic concept to remember is the pediatric inversion. Infants and young children naturally display an inverted neutrophil to lymphocyte ratio compared to adults. In pediatric samples, normal relative lymphocyte counts range high from 50 to 70%, while relative neutrophils drop to 20 to 40%. As pediatric patients mature into adults, this distribution shifts back to the adult pattern.
Next among the mature peripheral blood leukocytes is the monocyte, which holds the title of the largest white blood cell circulating in peripheral blood. Monocytes make up approximately 2 to 10% of total leukocytes in healthy adult blood. Morphologically, they are easily recognized by their abundant cytoplasm with a characteristic ground glass gray-blue hue, often containing fine azurophilic granules and phagocytic vacuoles. Their nucleus is highly variable, exhibiting an irregular, folded, or lobulated amoeboid appearance.
Monocytes perform two major physiological functions. First, they act as active phagocytes, essentially serving as cellular garbage collectors that clean up cellular debris, denatured proteins, and microorganisms. Once monocytes migrate out of peripheral blood vessels and enter tissue spaces, they further differentiate into tissue macrophages. Second, monocytes and tissue macrophages function as professional antigen-presenting cells. They internalize and process complex foreign proteins, displaying processed antigenic epitopes on their cell surface to educate lymphocytes and trigger targeted adaptive immune responses.
Moving to the granulocytes involved in specialized immune responses, we have the eosinophil. Eosinophils typically represent 1 to 3% of circulating peripheral blood leukocytes. Morphologically, they are characterized by a classic bilobed nucleus with 2 to 3 segments, but their defining feature is their cytoplasm, which is packed with coarse, uniform, reddish-orange specific granules. These granules contain basic proteins that selectively bind acidic dyes like eosin.
Functionally, eosinophils specialize in host defense against invasive parasitic infections, particularly helminth worms, by releasing cytotoxic granular proteins directly onto the parasite's surface. They also modulate immediate hypersensitivity and allergic reactions. To demonstrate how absolute counts function across different cell types, consider a patient with a total white blood cell count elevated to 20,000 per microliter, or 20 times 10 to the third per microliter, and an absolute eosinophil count calculated at 5,000 per microliter. Dividing 5,000 by 20,000 yields a relative percentage of 25% eosinophils, indicating significant eosinophilia.
The final mature leukocyte type found in peripheral blood is the basophil, which is the least abundant cell line in normal human blood, holding a relative adult reference range of 0 to 2%. Under the microscope, basophils display prominent, large, coarse dark blue to purple granules in their cytoplasm. These granules selectively absorb alkaline or basic dyes.
These dark granules are so dense and abundant that they often overlap and obscure the underlying multilobed nucleus. Basophils play a central role in mediating immediate hypersensitivity and systemic allergic responses. Upon activation, their granules rapidly degranulate, releasing potent chemical substances like histamine, heparin, and various vasoactive mediators that trigger tissue inflammation, smooth muscle contraction, and increased vascular permeability.
Having reviewed all 5 nucleated white blood cells, we turn to the non-nucleated cellular elements of peripheral blood, erythrocytes and thrombocytes. Mature human red blood cells, or erythrocytes, are unique because they lose their nucleus during terminal maturation. Morphologically, they circulate as flexible anucleate biconcave discs measuring 6 to 8 micrometers in diameter. Their primary physiological responsibility is transport, delivering oxygen from the lungs outward to peripheral tissues and carrying metabolic carbon dioxide waste from tissues back to the lungs.
Because oxygen transport is vital to cellular respiration throughout the human body, erythrocytes exist in vastly higher concentrations than leukocytes or thrombocytes. The normal adult reference range for red blood cells is 4.0 to 6.0 times 10 to the sixth per microliter, or 4.0 to 6.0 million cells per microliter. That means circulating red blood cells are present at concentrations roughly 1,000 times higher than white blood cells or platelets.
Lastly, we have thrombocytes, commonly known as platelets. Platelets are not intact whole cells, rather they are small anucleate cytoplasmic fragments shed directly into marrow sinuses by giant multinucleated megakaryocytes residing within the bone marrow. Platelets circulate at a reference range of 150 to 450 times 10 to the third per microliter, or 150,000 to 450,000 per microliter.
Functionally, platelets are essential for maintaining vascular integrity and initiating primary hemostasis. When blood vessel damage occurs, platelets rapidly adhere to exposed subendothelial collagen, undergo activation and aggregation, and form a primary platelet plug to stop acute bleeding.
Evaluating both the quantitative counts and morphological features of plasma, leukocytes, erythrocytes, and platelets give us the complete diagnostic picture we need to interpret hematology profiles effectively. Thanks for joining us, and we'll see you in the next review.